Emmaus. I'm the Science Director of Science Chicago. I think most of you are familiar with our program. I just want to take a quick minute and acknowledge our funders, the Sean D. and Catherine T. MacArthur Foundation, the Sorrel Funds of the Chicago Community Trust, Abbott, the Boeing Company, Illinois Toolworks, and Motorola. Science Chicago is spearheaded by the Museum of Science and Industry and is made possible by the collaboration of over 120 partners in the Chicago region, including universities, school systems, corporations, museums, the scientific community, and civic organizations. It's my pleasure to introduce tonight's speaker, Dr. Sean Decatur, who is the newest Dean of Arts and Sciences at Oberlin College in Ohio, where he's also Professor of Chemistry and Biochemistry. Before coming back to his home state earlier this year, Dr. Decatur spent 12 years at Mount Holyoke College, creating a world-class, use the microphone. This one? Do I have to start over? Okay. I'm a scientist. I am, so this is not, I'm sorry. Okay. Is it on? I turned it on. It's green? It sounds like it's on. Hello? Okay. Yes, okay. Before coming back to his home state of Ohio earlier this year, Dr. Decatur spent 12 years at Mount Holyoke College, creating a world-class research program in biophysical chemistry and engaging liberal arts undergraduates in scientific studies. He is well respected as both a scientist and an educator, receiving the Maribeth E. Cameron Faculty Prize for Scholarship, a Henry Dreyfuss Teacher Scholar Award, and a prestigious Faculty Early Career Development Grant from the National Science Foundation. Dr. Decatur's work focuses on the structure and function of proteins and how malformations in these proteins are linked to diseases like Alzheimer's and cataract, the latter being a focus of his talk this evening. He is also a strong advocate for the importance of undergraduate research and collaboration and is committed to promoting such interdisciplinary research at our nation's liberal arts colleges. I actually met Sean earlier this afternoon and had a wonderful time speaking with him about his work in Science Chicago and, of course, the upcoming election. And it's my pleasure to introduce him this evening for his presentation, Finding Clear Views of Blurry Lenses.
Thank you, Robbie, and thank you for coming out. Can everyone hear me without a mic? So I'm going to talk to you about today sort of two things that I am just incredibly passionate about. One which is a little bit odd, and that is proteins as molecules. So I have had an obsession with understanding proteins and how they work really for the past almost 20 years. And so I'm going to talk to you a bit about some of the work that's come out of this obsession. But in the process of doing that, I'm also going to tell you a bit of a story about undergraduate research, which is something else that I'm very passionate about. In the sense that, what I'd like to convince you of by the end of this evening is that you can really combine doing first-rate science and really first-rate education. That the way that we should be thinking about training not only our future generation of scientists, but I think actually students who are prepared to do problem solving in a wide range of different types of contexts, is one approach is to directly engage them with the production of new knowledge and getting them into the research lab and working on problems as early in their career as they can. So the work that I'm going to talk to you about today is really a project that had been working on at Mount Holyoke in working with undergraduate students there for the time that I was there, and we've actually brought some of this work with us to Oberlin since I came to Oberlin about 3 months ago. Mount Holyoke is a liberal arts college, a women's college, so you will see lots of pictures of women in my lab. I sort of went through many, many years of going to scientific meetings and having folks ask if you've ever worked with men. And it's well, sort of, but not often. But sort of an undergraduate women's college with a very diverse population of students. And so we'll actually get to see, as well as I go over the course of the talk, some of the actors who've been carrying out this research over the years. So the particular problem or application of my obsession with proteins I'm going to talk to you about today is cataract. Okay? Cataract is a disease which involves a fogging of the lens of the eye. This is a picture, actually, of a cataractus lens. Okay, and if you tell all of those actually have, if you look through, you can see this kind of clouding and the, basically the distortion of the type underneath. Okay? And this clouding is actually due to transformations of the proteins in the lens of the cell, in the lens of the eye. And so that's really the problem that I'm going to talk to you a bit about today. But first I want to take a step backwards and just kind of introduce you to the same obsession with proteins, again, that I've had for about 2 decades now. And this is a huge bias statement coming, but by far, if you think of the most important molecules that make up a living system, so the things which actually make all living systems possible, the things that make you what you are, proteins, if you take a strict reductionist viewpoint, you can come down to a protein molecule responsible for every aspect of a living system, ranging from the things which control and express genetic information to the things which are responsible for transmission of neurological signals, the things that are responsible for storage of information in the brain, generating energy. All of those actually have proteins as really the primary mechanism at the molecular level that explains those processes. So proteins are molecules. They're made up of units that are called amino acids. They're polymers, and so they're chains of these amino acids. And they're sort of these linear chains that are quite floppy in their shape once they're produced. And so it's almost like if you had sort of cooked spaghetti almost, okay? The sort of chains of these subunits that are these floppy long strings. What's remarkable is that each protein in your body, and it's coded by the genetic information that's inside the cell, folds into a unique 3-dimensional shape. And that 3-dimensional shape is responsible for the function that the protein does. So you have a protein that's responsible for each one of these jobs that have to happen inside the cell. And each one of those proteins has a unique shape. And actually, one of my favorite examples is this protein here. It's a protein called dihaloperoxidase. Okay? And it's an enzyme. It's an enzyme which does actually what sounds like on the surface a very non-biological thing. So enzymes are proteins which break down other molecules, and so they catalyze reactions. They catalyze different types of chemical reactions. And the chemical reaction that this catalyzes actually breaks down toxins, things called halogenated phenols, that are actually really very nasty things. And actually, some of them are produced by people often related to the paper industry and some bleaching of paper in paper processing. It turns out, though, if you go to the kind of mud flats on the Carolina coast, they're actually full of these halogenated phenols. And they're not actually there from human sources. They're there from natural marine sources that produce them. And they're produced in part by creatures that look like this. This is called the spaghetti worm. It actually really is this bright red color. And it's about, it's a worm, but can be quite long, up to 18 inches long. And sort of a quite thick little creature. If you were walking along the beach, you would not want to, and it wouldn't hurt you, but it would be sort of gross if you stepped on it. And it tends to bury itself in the soil, and these little tentacles stick out, and those kind of collect food as the tide sort of washes over the surface. And so it's bright red because it has this protein, dihaloperoxidase, in it. And what's interesting is that this worm actually lives in the mud flats, and it lives with other worms, other species of worms that live in the mud flats. And to defend itself against predators, these worms produce some of these nasty compounds called halogenated phenols. Okay? And they're bad for us. They're bad for other creatures as well that may want to eat the worms. And so they produce it and secrete it out into their environment as a defense mechanism. So basically, they kind of pollute their environment. They pollute their local environment against other types of predators. Of course, they can't kill themselves, so it would be a bad thing if they killed themselves. So these worms have an enzyme that's capable of breaking down this nasty toxin, the dihaloperoxidase. And what's remarkable is that this enzyme, it's very similar to other enzymes and other proteins. It's actually similar to proteins that are inside people as well.